In 2017, marine biologists working off the coast of South Africa started finding great white shark carcasses washing up on False Bay beaches. Not decomposed, not bitten apart. Each one had a single clean incision on its underside, and one organ missing, the liver, every single time. They pulled footage, reviewed tracking tags, cross-referenced sightings. The trail led to 2 male orcas. Researchers named them Port and Starboard, after their unusually collapsed dorsal fins, one leaning left, one leaning right. They moved together, and everywhere they went, the great whites left. Not just fled the attack site, left the entire region. Sharks that had been tagged and monitored in False Bay for years vanished from the data within days of Port and Starboard arriving. The sharks were not eaten. What Port and Starboard did was this. They would locate a great white, work it toward the surface together, and one of them would position itself beneath the shark and flip it upside down, a state called tonic immobility, where great whites go completely still and unresponsive. The other would then apply pressure to the body cavity from the outside, squeezing the carcass methodically until the liver slid out through the incision. They took the liver. They left the rest to sink. A single great white liver can weigh close to 600 pounds. It contains enormous concentrations of squalene, a lipid compound with one of the highest caloric densities found in any marine tissue. Port and Starboard had identified the most nutrient-rich component inside the largest apex predator in the ocean and developed a repeatable, efficient method to extract it with surgical consistency. Biologists confirmed the technique was not instinctual. It was learned, and it was precise. That alone would be enough. But the liver operation is almost the least interesting thing about orcas. Orcas, Orcanis orca, are the largest members of the dolphin family. Adult males can reach 32 feet and close to 12,000 pounds. They live in tightly bonded family groups led by matriarchs, older females who can survive past 90 years, and they are one of the only species on Earth where females continue living for decades after they stop reproducing. Researchers believe this exists because the knowledge those females carry is irreplaceable. They are libraries that move through the ocean. That knowledge includes hunting strategies, specific dialects, vocal patterns unique to each pod, incomprehensible to orcas from different groups, cultural behaviors transmitted from mother to offspring and maintained across generations. Scientists who study cetacean cognition place orcas among the most cognitively complex animals ever documented. There is currently no confirmed record of a wild orca fatally attacking a human being. That last part is worth holding on to. Not because it makes them less dangerous, because it means every interaction they have ever had with a human has been a deliberate choice. Off the coast of Norway, groups of orcas have been observed herding enormous schools of herring into tightly compressed balls near the surface, then striking the water with their tail flukes to generate concussive pressure waves, not to feed immediately, but to stun first. In Patagonia, researchers have filmed orca calves being brought to specific beaches by their mothers repeatedly, guided through the technique of intentional partial beaching to grab sea lions from the shoreline. It takes years to master. The mothers demonstrate. The calves practice. Getting it wrong at full commitment means getting stranded on the beach. The mothers teach the margin. This is not instinct. Instinct does not require years of supervised practice and graduated repetition. And then there is what happened between 2020 and 2023 off the Iberian Peninsula. A matriarch in a population that researchers had been tracking for years, designated white Gladys in the scientific literature, began approaching sailing vessels and engaging their rudders. Not the hulls. Specifically the rudders, the steering mechanism at the rear of the boat. She would bite and rotate until the rudder failed or snapped. Then other members of her group started doing it. Young orcas who had never done this before began replicating the behavior after watching it happen. Biologists do not use the word copycat in peer-reviewed papers. The term they use is cultural transmission of a novel behavior. It means exactly the same thing. Between 2020 and 2023, over 500 documented interactions with vessels were recorded along the Iberian coast. Several boats sank. No one has established with certainty what started it. Some researchers proposed White Gladys had a prior negative encounter with a vessel. Others suggested the behavior began as exploration. Orcas are known to investigate unfamiliar objects and escalated. What is not in dispute, the behavior spread deliberately through a population, was passed to younger individuals who had no prior context for it, and has continued. Orcas have no natural predators. They operate in every ocean on Earth. They communicate in ways we cannot fully decode, remember things we cannot fully map, and make decisions we cannot fully anticipate. The only reason the documented record of their behavior is not longer is that most of what they do happens underwater, out of reach, in places where we are simply not watching. Port and starboard were last documented in 2021, still moving together, still leaving great white carcasses on South African beaches with one organ cleanly removed. The great white population in False Bay, once one of the most stable and closely monitored in the world, has not recovered. Jewel wasp. There is an insect roughly the size of a paperclip, colored iridescent green and copper, that looks like it belongs inside a display case in a natural history museum. It lives across tropical regions of Africa, South Asia, and the Pacific Islands. It hunts cockroaches, and the method it uses took neuroscientists years to fully explain, because what it does should not, by any reasonable biological expectation, be possible. The emerald jewel wasp, Ampulex compressa, does not paralyze its target. Paralysis would be the simpler solution. A paralyzed cockroach stops moving, cannot resist, and begins to decay. That is useless to the wasp because what it needs is not a non-functioning cockroach. It needs a fully functional one that simply will not run. That distinction is the entire point. When a jewel wasp locates a cockroach, it delivers 2 stings in rapid sequence. The first targets the thorax and produces a brief, localized immobility of the front legs, just long enough to hold the cockroach in place for what comes next. The second sting goes directly into the cockroach's brain, not near it, into it. The wasp's stinger at this stage operates less like a syringe and more like a probe. It carries mechanosensory receptors at its tip that allow the wasp to detect pressure and texture from inside the tissue, meaning the wasp navigates through the cockroach's brain by feel, in real time, searching for a specific cluster of neurons called the escape ganglion. This is the structure that produces the decision to flee when a threat appears. The wasp finds it, deposits a chemically precise quantity of venom directly onto it, and withdraws. What follows is what neuroscientist Frederick Liebersaat spent years documenting in his laboratory in Israel. The cockroach does not run. It is not paralyzed. Its legs function. Its muscles respond. Its reflexes remain intact. Apply an electrical stimulus to a venom-injected cockroach and it walks. The body works. What it can no longer do is initiate movement on its own. The venom blocks octopamine receptors. Octopamine is the invertebrate equivalent of adrenaline, the compound responsible for triggering the impulse to flee when threatened. With the receptor blocked, the cockroach no longer generates the drive to escape, because the structure that produces that drive has been chemically switched off. The cockroach is present, aware in whatever sense a cockroach is aware, simply no longer motivated to act on any of it. The wasp then grabs the cockroach by the antenna and leads it. The cockroach follows. Liebersaat confirmed this through a series of controlled experiments. He introduced threatening stimuli to venom-injected cockroaches, vibrations, air pressure changes, physical contact. The cockroaches registered the inputs. They did not respond with avoidance behavior. In a separate experiment, he injected octopamine directly into the brain of a venom-treated cockroach. The cockroach recovered its escape response within minutes. The wasp had located one specific neurochemical switch inside an entire brain and turned it off, selectively, without disrupting anything adjacent. The wasp guides the now cooperative cockroach into a prepared burrow, attaches a single egg to its abdomen, and seals the entrance. The egg hatches in 2 to 3 days. Over the following 8 days, the larva feeds, and not randomly. Studies of larval feeding sequences have confirmed a consistent order of consumption that avoids organs whose early failure would accelerate decomposition of the surrounding tissue. The cockroach remains functional throughout the process. The larva completes its development and pupates inside the hollowed body, emerging eventually as a fully formed adult wasp. When toxicologists analyze the venom, They found it was not a broad-spectrum compound. It was a targeted pharmacological agent that crosses the blood-brain barrier, binds to a specific receptor subtype, and produces a precise behavioral change without causing systemic damage to the organism. The level of chemical selectivity involved, acting on one receptor class without disrupting adjacent systems, represents the kind of specificity that takes research teams years to engineer intentionally under controlled laboratory conditions. The jewel wasp developed it through evolutionary pressure, refined over millions of generations of contact with cockroach nervous systems. What Libersat's experiments made impossible to ignore is the detail that the cockroach was never absent from the experience. The capacity to move was present the entire time. The legs functioned. There was simply no instruction being issued. The wasp had not removed the cockroach's ability to act. It had removed the part that decides to. Butterflies. In the mid-1990s, an entomologist named Hans Benziger traveled to the forests of Thailand and Malaysia to prove something the scientific community largely considered implausible: that certain moths were capable of feeding on the blood of living mammals. Not as a biological accident, as a consistent, repeatable behavior. To confirm it, he sat in the jungle at night, rolled up his sleeve, and waited. The moth that landed on his arm was Calyptra thalictrae. It pressed its proboscis against his skin, the thin, tubular mouthpart that butterflies and moths typically use to draw nectar from flowers, and began to push. It did not find an existing wound, it produced one. The proboscis of Calyptra species has evolved a series of small, backward curved hooks along its surface, converting what was originally a drinking tube into something much closer to a barbed needle. The moth moved its head with short, forceful motions, working the tip through the outer layers of skin, and fed for approximately 20 minutes. Benziger photographed the entire process and confirmed the presence of blood in the moth's abdomen through laboratory analysis. The paper he published drew limited attention outside specialist entomology. It deserved more. The order Lepidoptera, butterflies and moths, contains roughly 180,000 known species. The vast majority feed on nectar, fruit juices, or plant fluids. The cultural image associated with them is almost uniformly one of gentleness. Weightless wings, flower visits, the transformation from something earthbound into something that flies. It is one of the most consistently positive associations any animal group carries in human culture across virtually every part of the world. That image is accurate for most of them. The ones it is not accurate for operate on a different principle entirely. It is called mud puddling, and the name makes it sound harmless. Butterflies gathering at puddles on the ground. The reality is that the sites they gather at are frequently not water. They are decomposing animal remains, feces, open wounds on living animals, skin soaked in sweat. Anything that concentrates sodium and amino acids, nutrients that flowers do not supply in the quantities certain species require. Male butterflies specifically are under significant reproductive pressure. During mating, males transfer to females a structure called a spermatophore, a protein-rich packet containing not only reproductive material, but a concentrated reserve of minerals the female uses directly in egg production. The quality of that packet, including its mineral content, determines in part how many viable eggs the female produces. Males with superior spermatophores leave more offspring. This is why approximately 90% of all butterflies observed at mud puddling sites are male. They are not wandering. They are loading inventory for a transaction that happens later. The specific sites they use reflect how intense this pressure is. Documented feeding locations include the carcasses of recently declined animals, the fluid from actively infected wounds on livestock, and the eye discharge of sleeping birds. The behavior scales to wherever the minerals are. And then there is lacrimophagy, feeding directly from the eyes of living animals. Researchers in Ecuador documented this extensively in field conditions. Butterflies and moths were observed landing near the eyes of river turtles and caimans, inserting their proboscises beneath the eyelids of the living reptiles, and feeding from their tear fluid. The insects returned to the same individual animals across multiple sessions. The fluid they targeted is rich in sodium, proteins, and lipids, exactly the compounds that nectar does not provide. Caimans, whose eyes sit at the waterline, were documented with multiple insects feeding from a single eye simultaneously. The reptiles showed signs of irritation. The insects finished before leaving. What Benziger's work, and the lacrimophagy studies that followed, confirmed is that the nutritional behavior of Lepidoptera exists on a wider spectrum than the standard image of them reflects. Most species never approach a wound or an eye, but those that do are not acting against their biology. The hooks on Calyptra's proboscis did not appear suddenly. They developed across vast spans of time in response to genuine pressure. Generations of moths encountering mammals and finding increasingly effective ways through. One final number. In populations where mud puddling is well documented, individual males visit multiple sites in a single day, accumulating mineral reserves that will be passed to a female who will convert them into eggs, which become caterpillars, which become the butterflies in the next season. The life cycle is exactly what it looks like. Egg, caterpillar, chrysalis, butterfly. What gets left out of that diagram is what the butterfly is doing between the flowers. Adelie penguin. In 1910, a British surgeon and naturalist named George Murray Levick traveled to Antarctica as part of the Terra Nova expedition, the same ill-fated mission led by Captain Robert Falcon Scott. Levick spent an entire summer on the ice observing a colony of Adelie penguins. What he witnessed disturbed him enough that he wrote his field notes in ancient Greek, a language the rest of the crew could not read. He called certain males gangs of hooligans. He noted behaviors he described as depraved and vicious. He compiled a report titled The Sexual Habits of the Adelie Penguin, submitted it to the Natural History Museum in London, and watched as the institution quietly shelved it. The report was considered too inappropriate for publication. It stayed out of public circulation for over a century. It was officially published in full in 2012. The behaviors Levick documented have since been confirmed by modern research. The Adelie penguin is not a complicated animal. It is a small, compact seabird that breeds on the Antarctic coastline, maintains a strict monogamous pair bond during breeding season, and builds its nests from one essential material: rocks. This is not a preference. In Antarctica, where the ground is frozen solid and vegetation does not exist, rocks are the only available insulation between an egg and the ice. A nest built from more rocks keeps eggs warmer. Warmer eggs produce more surviving chicks. Rocks, in an Adelie colony, are the closest thing to currency. And like any place where a resource is scarce and essential, the colony has its criminal element. Female Adelies have been observed leaving their nests, crossing into neighboring territory, and approaching unattached males. Males whose partners are away hunting. The female performs the courtship behavior the male expects: the lowered head, the particular posture associated with receptivity. The male responds. The female lingers just long enough and then, during the interaction, quietly transfers a rock from his nest to hers. Then another. She returns to her own nest with the material, and the male, if he pieces it together at all, is left with a slightly less insulated nest and no recourse. This is not a rare occurrence. Researchers observing Adelie colonies have documented it as a structured pattern of behavior repeated consistently across breeding seasons. Some females have been observed visiting multiple males in a single day, each time extracting rocks with the same approach. The pairs they target are not random. Lone males with well-stocked nests are selected specifically. The rock economy creates a secondary layer of behavior that Levick was among the first to document systematically: the vagrant males. Not all males in a colony maintain a nest or a bond. Some wander the edges of the breeding ground without a clear function in the reproductive structure of the colony. Levick observed these individuals behaving in ways he found shocking enough to require encoding. Modern researchers, working without Victorian-era moral frameworks, have documented what Levick saw without euphemism. Vagrant males in Adelie colonies engage in forced interactions with females, with injured individuals, and with those that have not survived the season. The latter category, interactions with non-living colony members, is not an occasional aberration. It has been documented consistently across field studies, involving individuals that have been inactive for extended periods of time, in some documented cases for more than a year, sometimes found in frozen and incomplete states on the colony margins. The reason it happens is the same reason everything in an Adelie colony happens: the rock instinct. The neurological drive to reproduce is, in some individuals, not connected cleanly to the conditions that make reproduction viable. The signal fires regardless of context. The result is what Levick encoded and what biologists have since observed on camera in normal field conditions without any attempt at concealment. The Adelie penguin is not doing any of this out of malice. It has no concept of what any of it means. That is, in some ways, the more unsettling part of the whole picture. The colony functions in its own logic, driven entirely by pressure, instinct, and access to small stones, and it produces a social environment that a Victorian surgeon found too graphic to publish in plain English. He was not wrong to be disturbed. He was just 100 years early. Indicator. There is one wild animal on Earth that actively communicates with humans to guide them toward a resource, not as a trained behavior, not in captivity. Spontaneously, in the field, as a strategy it developed independently over evolutionary time. The greater honey guide, indicator, searches out human beings, specifically the Yao people of Mozambique and similar communities across sub-Saharan Africa, produces a specific call to signal its presence, waits for a response, and then leads whoever answers through the bush toward a beehive. The human opens the hive, takes the honey, and leaves behind the beeswax and larval comb, which the honey guide needs but cannot access on its own. Both parties gain something. Neither deceives the other. It is the most frequently cited example of spontaneous cooperative communication between a wild animal and a human being. It appears in biology textbooks, nature documentaries, and educational material about cross-species relationships. Researchers have published extensively on it. The signal response system has been studied in detail and confirmed to be genuinely bidirectional. The birds distinguish between human calls and animal sounds, and they respond specifically to the traditional signals used by the Yao, with measurably higher cooperation rates than when other sounds are used. The greater honey guide is, by that measure, the animal that comes closest to understanding the concept of working together with us. It is also an animal that begins its life by systematically eliminating every other living thing in whatever nest it hatches in. The female honey guide does not build a nest or raise her own young. She lays her eggs in the active nests of other bird species, primarily bee-eaters and kingfishers, species that nest underground in burrows, in complete darkness. She watches a target nest over time, learns the schedule of its occupants, and moves during a window of absence. She removes one of the host's eggs, swallowing it or discarding it, and replaces it with one of her own. The substitution takes seconds. The honey guide egg hatches before the host's eggs. The chick that emerges is blind, featherless, and entirely pink, a small, soft mass of undeveloped tissue in a dark underground tunnel. It has no visual information about its environment. It cannot regulate its own temperature. It cannot feed itself. What it can do, from the first hours after hatching, is locate the other eggs in the nest by movement and warmth, grip them with its bill, position them against its back, and use a specialized hook on the tip of its beak, a sharp, curved structure present only in the first days of life and shed once its purpose is complete, to puncture and destabilize the shells until the eggs no longer develop. Any chicks that hatch before the honey guide can eliminate the eggs are dealt with through direct, repeated strikes until they stop moving. The BBC documented this process with infrared cameras placed inside active burrow nests in sub-Saharan Africa. The footage shows a newly hatched honey guide chick, no larger than a thumb, eyes sealed shut, no feathers, systematically working through the contents of the nest over a period of hours. There is no ambiguity about what is happening. The movements are deliberate and methodical. The chick that emerges from this process as the sole occupant of the nest will be fed, warmed, and raised by two birds that are not its parents, species that are smaller than it will eventually become, working themselves to exhaustion to raise something that has no biological relationship to them. None of this is hidden behavior. The adults that raise the honey guide chick can see what it is doing. Researchers have debated at length why host birds do not intervene more effectively. The answer appears to involve a combination of the darkness of the burrow, which limits visual information, and the fact that the honey guide chick, almost immediately after clearing the nest, begins producing a begging call that closely matches the acoustic profile of the host species' own chicks. It communicates in a voice that sounds like the offspring the hosts expected to have. The bee cook that makes all of this possible is gone within the first few days. It falls off on its own with no remaining trace once it is no longer needed. It is an adaptation with a single function in the entire lifespan of the animal, to ensure that nothing else survives to compete for the food that is coming. The same bird, as an adult, guides humans through the forest to honey. The cooperation it offers is real. The partnership is genuine. It just started with a series of actions that the host family, nesting in the dark below the ground, never fully understood. Cuckoo bird. The sound most people associate with the common cuckoo is from a clock. The cuckoo clock, invented in the Black Forest region of Germany in the 18th century, uses a two-note mechanical call to announce the hour, and that sound has become so embedded in European and global culture that it carries connotations of reliability, rhythm, domesticity. It is the sound of a house that works. The actual bird the clock was modeled on spends its life dismantling the houses of others. The common cuckoo, Cuculus canorus, is a brood parasite, one of the most sophisticated examples of biological deception documented in any species. The female does not build a nest. She does not incubate eggs. She does not feed chicks. She spends the breeding season watching other birds, identifying suitable targets, and executing a replacement operation that takes less time than it takes to read this sentence. The approach is precise. A female cuckoo selects a host species, typically a reed warbler, dunnock, or meadow pipit, depending on the lineage she belongs to, and monitors that nest for weeks before acting. She learns the host's routine: when both parents leave, how long they are gone, how many eggs are already present. When the window opens, she lands at the nest, swallows one of the host's eggs in a single motion, and deposits one of her own in its place. The entire visit lasts approximately 10 seconds. She does not return. What makes this operation function is the egg. A female cuckoo produces eggs that replicate with remarkable fidelity the visual appearance of the eggs laid by her target species. The coloration, the patterning, the size, the surface texture, all of it is calibrated to match. This is not a general approximation. Different cuckoo females, which tend to parasitize the same host species their own mothers used, produce eggs that are specifically matched to that species. Reed warbler cuckoos produce eggs that pass inspection in reed warbler nests. Dunnock cuckoos produce eggs that pass inspection in dunnock nests. The calcium glands that produce the eggshell are the mechanism. The pigment composition and deposition patterns are genetic, heritable, and specific enough that researchers have been able to identify which host species a given female's lineage has been targeting based solely on the characteristics of unhatched eggs found in museum collections. The cuckoo egg hatches earlier than the host's eggs, typically within 12 days, compared to 13 to 14 for most hosts. The chick that emerges is pink, blind, and ungainly. It is also, from the first hours of life, driven by a compulsion that overrides every other behavior. It pushes things out of the nest. The mechanism is well documented and straightforward. The chick backs against any object, egg or hatchling, braces itself against the nest wall, and uses its wings and legs to maneuver the object onto its back, then lifts and tips it over the edge. It does this repeatedly, systematically, until the nest contains only itself. A newly hatched cuckoo chick can shift an object 2 to 3 times its own body weight in this way. The instinct is strongest in the first 4 days and gradually diminishes after the nest is clear, but the physical capacity for it remains for some time. The host parents, watching this happen, do not intervene. Behavioral research has proposed several explanations. The chick produces a begging call at an intensity and frequency that overwhelms the parental feeding response, effectively locking the adults into a feeding loop regardless of the visual context. By the time the cuckoo chick has grown large enough to make the substitution obvious, often sitting in a nest that cannot physically contain it, surrounded by parents that reach only its shoulder, the behavioral override is complete. The parents continue bringing food. The most complete picture of what host species actually experience came from an experiment designed to test what researchers had termed the mafia hypothesis. The theory proposed that cuckoos do not simply parasitize host nests passively. They monitor them after laying and respond to rejection. To test this, researchers working with magpie nests artificially removed cuckoo eggs after they had been laid. The result was consistent. The female cuckoo that had placed the egg returned, located the nest from which her egg had been removed, and destroyed the nest, puncturing the remaining eggs and dismantling the structure. The magpies whose nests were destroyed rebuilt and began a new clutch. In several documented cases, the same female cuckoo parasitized the replacement nest within days. The behavior confirmed what the hypothesis predicted. A host that rejects the cuckoo's egg loses its current clutch. A host that accepts it loses its current clutch more slowly. Either way, the cuckoo's genetics continue. The host's options, practically speaking, are limited to which version of the outcome they prefer. The clock on the wall was modeled on a bird that does exactly this to whatever it lands in. It announces its presence in 2 clean notes and is gone before anyone has time to process what just happened. Dolphin. The bottlenose dolphin cannot stop smiling. Not because it is happy. The upward curve of its jaw is a fixed anatomical feature, a structural consequence of the way the skull is shaped, present in every individual regardless of circumstance. A dolphin in distress has the same expression as a dolphin at rest. The face communicates nothing about what is happening behind it. This has been historically a significant problem for public perception. The bottlenose dolphin, Tursiops truncatus, is one of the most cognitively studied animals on the planet. It passes the mirror self-recognition test, a benchmark used to assess self-awareness, reliably and repeatedly. It uses tools. Certain populations off the coast of Western Australia carry marine sponges on their rostrums while foraging along the seafloor, a behavior that is learned, passed from mother to offspring, and maintained as a distinct cultural tradition within specific groups. It has signature whistles, unique acoustic identifiers that function as individual names, recognized and used by other dolphins to call specific individuals. Researchers have documented dolphins responding to their signature whistle being played even years after last contact. The intelligence is not in question. What it produces, however, is more complex than the popular version allows. Bottlenose dolphins organize themselves into nested alliance structures that researchers classify into first, second, and third-order alliances. A first-order alliance is a small group of males, typically 2 to 3 individuals, who coordinate closely to pursue and maintain access to a single female. A second-order alliance is a coalition of multiple first-order groups that cooperate with each other in competition against rival coalitions. A third-order alliance is a broader defensive arrangement between second-order coalitions. The structure requires each individual to track relationships, obligations, and histories across dozens of animals simultaneously. For reference, human beings operate comfortably within social networks of roughly 150 individuals before the cognitive load becomes unwieldy. Dolphins and populations with multi-tier alliance structures are managing a social map of comparable complexity in an environment with no fixed landmarks, no written records, and constant movement. What the alliance structure is used for is not cooperative hunting. It is primarily the coordinated pursuit and confinement of females. Groups of allied males in Shark Bay, Australia, have been documented surrounding single females in open water, herding them and preventing departure, sometimes for weeks at a time. If a female attempts to leave the group, the males chase and physically redirect her. The behavior has been observed across multiple populations and confirmed by years of field research as a consistent feature of bottlenose dolphin social structure in areas where it has been studied in depth. Then there are the strandings. In the early 1990s, marine mammals began washing up in unusual numbers on the beaches of Moray Firth in Scotland and separately along the Virginia coast in the United States. The pattern was unusual. The animals were not fish. They were harbor porpoises, small, quiet cetaceans that share the same coastal waters as bottlenose dolphins, but occupy a different ecological position and pose no competitive threat to dolphin feeding. The carcasses showed severe blunt force trauma, fractured ribs, ruptured organs, hemorrhaging consistent with repeated high-impact strikes. There were no bite marks suggesting predation. Nothing had been eaten. Researchers initially considered military sonar testing as a possible cause. Autopsies eliminated that explanation. What the examinations found instead were tooth rake marks that matched, geometrically and spatially, the jaw structure of bottlenose dolphins. Follow-up field observation confirmed what the evidence had pointed toward. Local dolphin groups were pursuing and striking harbor porpoises with their rostrums and flukes, in groups, with the coordinated timing their alliance structure makes possible, and then leaving the animals on the seafloor or surface without any feeding behavior whatsoever. The behavior has no clear resource-based explanation. Porpoises do not compete meaningfully with dolphins for food. They do not threaten dolphin young. They occupy a different tier in the ecosystem. The working hypothesis in the research literature is that the behavior is a form of highly stimulated social play that escalated in intensity, that the same coordination mechanisms the dolphins use in hunting and alliance competition are being applied in a context with no survival payoff. Some researchers have noted parallels with the way juveniles in cognitively complex species practice skills in non-survival contexts. What those harbor porpoises experienced, framed that way, is the consequence of being in the water, when a group of highly coordinated, highly motivated animals needed something to do. The last detail that rounds out the picture came from BBC film crew footage reviewed by National Geographic researchers. The footage documents a group of bottlenose dolphins locating a pufferfish, agitating it gently until it released a quantity of tetrodotoxin, the potent compound pufferfish produce as a defense, and then passing the fish carefully between individuals, each one holding it briefly near the surface before transferring it to the next. After the circuit was complete, the group floated near the surface in what researchers described as a trance-like state, apparently unresponsive to normal stimuli for a period of time before resuming normal behavior. Tetrodotoxin in small, non-lethal doses produces altered neurological states. The dolphins were using the fish as a shared chemical experience. Whether this constitutes anything like what the word recreational implies in a human context is genuinely unknown. What is known is that they found it, knew what it did, handled it carefully enough not to receive a harmful dose, and shared it in a deliberate sequence. The smile on their faces throughout all of this was, as always, completely uninformative. Amazon ant. Somewhere in a temperate forest in North America or Europe on a warm summer afternoon, a column of ants is moving through the undergrowth. The column can contain 2,000 individuals or more. They are moving quickly, in formation, and they are not foraging. There is no food at their destination. There are no resources they are trying to protect. They are moving toward another colony of ants with a single objective: to take everything that colony will ever produce. The Amazon ant, Polyergus spp, is an obligate slave maker. The word obligate in this context means that the behavior is not optional or situational. It is the foundation of the species' survival. Amazon ant colonies cannot function without the labor of other species. They do not gather food. They do not tend larvae. They do not construct or repair the nest. They are incapable of performing any of these functions, not because they choose not to, but because their bodies have been shaped by evolution into tools for a single purpose: raiding. The mandibles of the Amazon ant are long, curved, and razor-sharp, shaped like sickles. Under a microscope, the cutting edge is smooth and continuous. There is no serration, no gripping surface, nothing designed to hold or manipulate an object carefully. These mandibles are extraordinarily effective at piercing and restraining other insects. They are completely useless for handling food. An Amazon ant placed next to a food source it needs to survive will go hungry before it figures out how to feed itself. Researchers have confirmed this in laboratory conditions. Isolated Amazon ants placed alongside abundant food sources starve without a slave worker present to process and transfer the food into their mouths. The colony sustains itself through raids conducted against neighboring colonies of Formica ants, smaller generalist workers who build nests, forage, tend brood, and perform all the domestic functions a colony requires. The Amazon ants march to a Formica nest, overwhelm its defenses through speed and numbers, locate the developing pupae, the immature ants still in their cocoons, and carry them back. The Formica larvae and eggs are generally left behind. What the raiders want is specifically the pupae, because pupae taken to a foreign nest and allowed to complete development will emerge already chemically oriented to their new environment. They will not know they are in the wrong colony. They will work. The chemical component of this is central to how the whole system operates. Ants identify their colony mates through a suite of cuticular hydrocarbons, chemical compounds on their body surface that function as a colony-specific chemical signature. Pupae that develop inside an Amazon ant colony absorb the chemical profile of that environment during the final stages of development and emerge treating that profile as home. There is no detectable transition, no period of confusion or adjustment. The formica workers that hatch in an Amazon nest simply begin working. Without a constant supply of new pupae, the slave population ages and shrinks. When it drops below a functional threshold, the colony cannot sustain itself. This is why the raids are not occasional. They are scheduled. Some colonies conduct raids multiple times per week throughout the active season, and the scale can be significant. A single raid has been documented removing several thousand pupae from a target nest in a single operation. The founding of a new Amazon colony follows the same logic, taken to its most direct form. A mated queen locates an active formica nest, enters it alone, and proceeds to the brood chamber where the resident queen is located. She engages the resident queen directly and eliminates her, a process that can take up to 30 minutes of sustained contact. She then does something that researchers describe as one of the more remarkable chemical behaviors documented in social insects. She coats herself in the chemical signature of the queen she just removed. She rolls across the body. She absorbs the cuticular hydrocarbons. She becomes, chemically, the colony's queen. The formica workers whose queen just stopped living do not react with aggression to the Amazon queen standing in the nest. They react with recognition. The chemical signal they receive matches the signal they were oriented to. Within seconds of the transition, workers that had been defending the nest begin grooming and feeding the intruder. The colony continues as if nothing changed. A new queen simply appeared, and the old one is no longer present. The colony that results from this founding will produce no new Amazon workers of its own, without further raids. The workforce it has is the workforce it took. When those workers age out, more must be taken to replace them. The empire is built entirely on continuous acquisition, and the moment the acquisition stops, the empire collapses from the inside. Baboons. In the early 2010s, conservation managers on the Cape Peninsula in South Africa began receiving an unusual volume of incident reports about a single individual. The subject was male, large, and had developed a skill set that was causing specific problems for the local population. He had learned to open car doors. His name, in the management records, was Fred. Fred was not operating alone. He led a troop of baboons that had become highly adapted to the suburban and semi-urban environment of the Cape Peninsula, an area where human residential neighborhoods, tourist infrastructure, and wildlife corridors exist in close proximity to one another. The troop's behavior had evolved well beyond foraging at the edges of human spaces. They moved through neighborhoods with a level of environmental familiarity that conservation officials found difficult to manage. Fred's specific contribution was tactical. He had understood through observation and trial that cars contain food and that cars can be opened. He applied this understanding systematically. Reports described a consistent pattern. While other members of the troop created a visible disturbance at the front of a vehicle, drawing the attention of any occupants or bystanders, Fred would approach from the rear, work the door handle, and access whatever was inside. The coordination was not coincidental. It was repeatable across multiple incidents and with the same general structure. This is not unusual for baboons as a species. The olive baboon and the chacma baboon have both adapted extensively to human-adjacent environments across sub-Saharan Africa, developing behavioral repertoires that reflect direct observation and learning of human routines. They know when market days occur. They know which windows in which buildings are typically left open. They know the schedules of the people who feed them and the people who chase them, and they respond differently to each. Field researchers working with baboon populations in Kenya and South Africa have documented individuals that can distinguish between armed and unarmed rangers and adjust their behavior accordingly, retreating from uniformed personnel carrying rifles while continuing to approach unarmed tourists. The social structure that supports this behavior is one of the most complex among non-human primates. Baboon troops are organized around a dominance hierarchy maintained by ongoing negotiation, alliance formation, and, when necessary, direct physical confrontation. Males at the top of the hierarchy carry canine teeth, the elongated upper fangs, that can exceed 5 centimeters in length. For context, the canines of an adult male lion average approximately 7 centimeters. A male baboon's canines, proportional to its body size, represent a comparable commitment of biological resources to a weapon, and the baboon deploys them with the full awareness of what they signal. The threat display, lip curling to expose the full length of both canines while producing a low vocalization, is something that registers at a very deep level in the human nervous system, which makes evolutionary sense given how long the two species have been in proximity to each other. Fred's troop was generating enough property damage and enough incidents of physical aggression toward residents and tourists that the local government designated him a danger to public safety. Conservation teams had already attempted relocation, a standard management response to problem individuals, more than once. Fred had returned each time, demonstrating the spatial memory and navigational capacity that researchers have documented extensively in wild baboon populations. He knew where he was and how to get back to where he wanted to be. The management decision that followed was official, documented, and formal. A lethal control order was issued for a single baboon, signed off by a government body because no other available tool, relocation, hazing, barriers, had proven sufficient to interrupt his activities. Fred was found and euthanized in 2011. The Cape Peninsula Baboon Management Joint Task Team, which oversees the roughly 16 troops active in the region, now employs dedicated baboon monitors who follow the troops daily, using paintball guns and noise deterrents to maintain boundaries between baboon ranging areas and residential zones. The program runs year-round. It requires trained personnel, equipment, vehicles, and a response protocol. It is funded by local government and operates as a permanent feature of municipal services in the area. This is the infrastructure that a population of animals that weighs, on average, about 44 pounds, has made necessary. In Saudi Arabia and parts of East Africa, hamadryas baboons have been documented doing something that researchers at first treated with some skepticism before the behavioral evidence accumulated to a point where it could not be dismissed. Male baboons in these populations had been observed capturing very young dogs, puppies, typically feral or isolated ones, carrying them alive back to the troop and keeping them. The dogs were not eaten. They were held, intimidated into submission through repeated aggressive contact, and eventually incorporated into the troop's daily movement as animals that traveled with the group and responded to it. Over time, researchers noted that the dogs were reacting to external threats in ways that aligned with the troop's interests. Dogs that attempted to leave were retrieved. Dogs that had been with the troop for extended periods showed behavioral indicators of orientation toward their captors rather than away from them, responding to baboon vocalizations, remaining in proximity voluntarily, and in several documented cases, reacting aggressively toward unfamiliar dogs that approached the troop. The researchers who published in Animal Behavior on this subject used careful language. The dogs, they noted, showed indicators of what could be described as a stress-conditioned attachment bond, a behavioral state in which an animal that has no viable path to independence and has been conditioned through sustained stress to associate proximity to the source of that stress with a reduction in that stress. The dog does not protect the baboon because it wants to. It protects the baboon because it does not know what else to do. Fred would have recognized the logic immediately. Sea otter. There is a video that has been shared millions of times across the internet. 2 sea otters floating on their backs in calm water, holding hands. They drift together slowly, maintaining contact, and the caption that almost always accompanies the clip explains that they do this so they don't float apart while sleeping. The video generates a specific kind of reaction, a warm, uncomplicated one, and it has done a great deal to establish the sea otter as one of the more beloved marine mammals in popular culture. The behavior in the video is real. Sea otters do hold hands while resting. It is called a raft, and it does help individuals stay together in current. Everything else about the sea otter requires a completely different conversation. The sea otter, Enhydra lutris, lives in the cold nearshore waters of the North Pacific, from California up through Alaska and across to the Russian Far East. It is the heaviest member of the weasel family. It has the densest fur of any mammal on Earth, up to 1 million hair follicles per square inch. Because it has no blubber and relies entirely on that fur layer to maintain body temperature in water that sits just above freezing. To sustain its metabolism in those conditions, a sea otter must consume approximately 25% of its body weight in food every single day. In Monterey Bay, that means diving repeatedly for sea urchins, clams, crabs, and abalone, often using rocks as tools to crack open shells at the surface. This is the life of the female sea otter. The male sea otter, particularly in populations where adult males outnumber available females, has developed an additional behavioral strategy to ensure reproductive access. It involves the pups. When a female sea otter is accompanied by a young pups that are too buoyant to dive and depend entirely on their mothers for food and warmth, certain males have been documented approaching the pair, seizing the pup, and holding it below the surface. Not to harm the pup directly, to hold it there, just below the surface, while the mother reacts in distress, attempting to recover her offspring. The male holds the position until the female, who needs to surface repeatedly herself, who is panicked, who has no alternative, hands over the food she has just spent energy diving for. The male releases the pup, takes the food, and moves on. This behavior has been recorded across multiple sites and confirmed through underwater observation and tagging data. It is not a one-time aberration by a single individual. Researchers have documented it as a repeatable pattern in populations where male-to-female ratios create high competition for mating opportunities. The calculus from the male's perspective is straightforward. The female needs the pup to survive. The male controls access to that outcome temporarily. The female pays. In 2010, biologist Heather Harris and colleagues at the California Department of Fish and Game published a report that extended the documented behavioral picture considerably further. Researchers had been finding the bodies of harbor seal pups on beaches around Monterey Bay. Small individuals showing signs of injury inconsistent with predation or stranding. The investigation traced the source of these incidents to specific male sea otters in the area who had been observed approaching harbor seal pups that had been left on the beach by their mothers, a normal part of harbor seal pup development, during which the mother leaves to feed and the pup waits on shore, and engaging them in sustained forcible contact that left the pups non-viable. The behavior had no feeding component. The pups were not consumed. They were not competitors for food. They were, by the available evidence, substitutes, interactions driven by the same biological pressure that shapes all male sea otter mating behavior, redirected toward a target that posed no resistance. The population-level data from Monterey Bay adds a final number. Approximately 11% of documented female sea otter fatalities in the area are attributed to trauma sustained during mating interactions with males of their own species. In cold, calm water, with no external predators present, roughly 1 in 10 female sea otters does not survive the reproductive process. The hand-holding happens too. Both things are true at the same time. Meerkat. Stand at the edge of a meerkat colony in the Kalahari at sunrise, and you will see exactly what the documentaries show. Small, upright figures facing the morning sun, warming themselves after the cold of the desert night. The eldest individuals posted on high ground scanning for aerial threats while the younger ones forage below. It is coordinated, cooperative, and visually striking. It is also the surface layer of a social structure that, once examined in detail, does not resemble cooperation in any meaningful sense. The meerkat, Suricata suricata, lives in tight family groups called mobs, typically ranging from 3 to 30 individuals in the arid regions of southern Africa. Each mob is dominated by a single breeding pair, the alpha male and alpha female, who produce the overwhelming majority of the group's offspring. The other adults in the group, most of them related to the dominant pair, function as helpers. They babysit, they sentinel, they bring food to pups that are not their own. The system is frequently described in introductory biology as a model of cooperative breeding. What it does not typically describe is how the alpha female maintains her position. The dominant female in a meerkat mob produces chemical signals, secreted through a scent gland located near the base of the tail. that she distributes through the group by rubbing against subordinates and marking shared spaces within the territory. These secretions are not just identifying markers. Research published through the Kalahari Meerkat Project, one of the longest-running continuous studies of a wild animal population in the world, has documented that the chemical compounds in the dominant female's secretions interact with the endocrine systems of subordinate females in the group, suppressing the hormonal signals that would normally trigger ovulation. The dominant female does not need to prevent subordinate females from mating through physical intervention. Her chemical presence does it passively. Females that live within scent range of a dominant individual cycle irregularly or not at all. Those that are removed from the group, relocated in experimental conditions, resume normal reproductive cycling within a short period. Return them to the mob and the suppression resumes. This is not incidental. It is the mechanism by which the dominant female concentrates reproduction in herself. The other adult females in the group are metabolically capable of producing offspring. They are physiologically prevented from doing so by the chemical environment the dominant female creates by existing in proximity to them. When the mechanism fails, when a subordinate female becomes pregnant despite the suppression, the response is direct. The dominant female will seek out and eliminate the litter, often consuming it on the site. This behavior has been confirmed through camera trap footage, direct observation, and post-mortem analysis of subordinate females' reproductive histories within the long-term Kalahari dataset. The dominant female does not consistently act against litters produced by unrelated individuals encountered outside the group. The targeting is internal. It is family. The Kalahari Meerkat Project records extend further. In cases where a subordinate female survives the elimination of her litter, she does not simply return to non-reproductive helper status. She frequently enters a state of physiological lactation triggered by the stress of the event, producing milk without having live pups to feed. The dominant female then directs her own pups toward the subordinate female, who nurses them. The subordinate female, having had her offspring removed, feeds the offspring of the individual responsible. Subordinate females that become pregnant and manage to avoid losing their litters face a separate consequence: eviction. The dominant female expels them from the group, forcing them to navigate the Kalahari without the sentinel system, the shared warmth of communal sleeping, or the predator alarm calls that make group living survivable in a landscape that contains eagles, cobras, and jackals. Alone in the Kalahari, a meerkat's life expectancy drops sharply. The eviction is not issued with that intent, but the outcome is consistent. The final number from the long-term data is the one that anchors all of this in a broader context. Across the population monitored by the Kalahari Meerkat Project, researchers calculated that approximately 20% of all meerkat fatalities in the wild are the direct result of intentional acts by other meerkats, not predation, not accident, not disease, meerkats. No other mammal species has been documented at a comparable rate of within-species fatal conflict relative to its total mortality. Not wolves, not lions, not chimpanzees. The animal that has been used in children's educational material to illustrate the concept of community responsibility holds, in the ecological literature, a record that no other mammal comes close to matching. Praying mantis. The praying mantis gets its name from its posture. At rest, the forelegs are folded inward and held close to the body, elevated slightly in a position that resembles, to the human eye, a figure at prayer. The comparison has attached itself so firmly to the animal that it appears in its common name in dozens of languages, and cultures across Asia and the Mediterranean have historically attributed to it qualities of patience, mindfulness, and spiritual attentiveness. The Chinese martial arts tradition includes a fighting style modeled directly on its movements. The reverence is understandable. The mantis is visually striking, genuinely patient in its stillness, and moves with a precision that is difficult not to read as intentional. What the posture actually represents is a weapon system in standby mode, and what happens when it activates has very little to do with contemplation. The forelegs of the praying mantis, Mantis religiosa, and related species are called raptorial appendages. They are not arms in any functional sense analogous to a vertebrate limb. They are closing mechanisms. The inner surfaces of both the upper and lower segments are lined with sharp, inward-facing spines. arranged so that when the two segments snap together, whatever is between them is gripped from multiple angles simultaneously and cannot be pulled free without moving against the direction of the spines. The joint that drives the closure is powered by a fast twitch muscle arrangement that operates in approximately 30 milliseconds, fast enough that high-speed cameras are needed to resolve the motion. The mantis does not grab prey. It closes a trap around it. The strike range is precisely calibrated. Mantids spend the majority of their active time absolutely still, tracking moving targets with their eyes. They are among the few insects capable of true stereo vision, able to perceive depth, and they do not strike until the target enters a specific distance window. Outside that window, they wait. At the threshold, the forelegs extend and close before any vertebrate nervous system registers that movement has begun. What happens after the capture is where the mantis's profile for this particular list becomes relevant. Female praying mantids are larger than males, significantly so in many species, sometimes by a factor of 2 or more. The size difference exists in part because larger females produce larger egg masses, and egg mass size correlates with reproductive output. The female's nutritional requirements around the time of mating are high, and she is a predator. The male is present. During mating, in a proportion of documented encounters across multiple species, the female turns and consumes the male's head. Not after mating. During it, the male continues. This is the part that initially seemed implausible and has since been studied in enough detail to be well understood mechanistically. The head of the male mantis contains the subesophageal ganglion, a nerve cluster that, among its functions, sends inhibitory signals that moderate the pace of the reproductive process. When the female removes the head, those inhibitory signals stop. What researchers have measured is that decapitated males in mating context show increased intensity and duration of the copulatory behavior compared to intact males. The removal of the head does not interrupt reproduction. It removes the neurological governor on it. The female gains the nutritional content of the male, which is substantial relative to her body mass, and a maximized reproductive output from the mating event. The male's body, operating without central coordination but with intact spinal reflexes, continues through the process to completion. Biologist William Brown of the State University of New York has studied mating behavior across mantis species in both field and laboratory settings. His work confirmed what the physiological evidence suggested. Females that consume their mates during copulation produce measurably larger egg masses than females that mate with males that survive the encounter. The difference is not marginal. The consumption translates directly into more eggs, and more eggs means more surviving offspring in the next generation. The biology does not frame this as a loss. From a genetic standpoint, the male's contribution is higher in this scenario than in the scenario where he survives. His genes appear in more eggs. The cost is specific to him and considerable, but the accounting is not done from his perspective. What Levick found disturbing enough to write in Greek, what Libersat confirmed in a laboratory with electrodes, what Heather Harris documented in cold water off Monterey Bay, all of it is the same underlying principle. The biological machinery that produces behavior runs without any mechanism for evaluating what that behavior looks like from the outside. It runs because it works. The mantis does not know what prayer is. The posture just holds the forelegs in the best position for what comes next. Chimpanzee. Jane Goodall had known Goliath for years. He was one of the oldest males in the Gombe community, large, familiar, a figure she had observed through years of fieldwork in Tanzania. He had been a dominant individual in his prime. By 1974, he was older, less central to the group's dynamics, but still a recognized member of the Kasakila community. In 1974, members of the Kasakila community found him alone. They held him down and did not stop until he was no longer moving. Goodall later said that what happened at Gombe between 1974 and 1978 gave her nightmares for years. Not specifically because of the violence. She had witnessed aggression in chimpanzee communities before. What disturbed her was the sustained, deliberate nature of what unfolded, and the fact that the individuals doing it had history with their targets. Goliath was not a stranger. He was known. The distinction did not matter. The chimpanzee, pan troglodytes, shares approximately 98.7% of its DNA with human beings. This figure is cited frequently in biology education as a reminder of evolutionary proximity, usually in the context of cognition, tool use, or social complexity. It is equally applicable to the behavioral repertoire that played out at Gombe. What happened there over 4 years was documented by Goodall and her research team, and has since been analyzed extensively as the first confirmed observation of what researchers classify as intergroup lethal rating in a non-human primate. The Kasakila community and the Kahama community, the latter a splinter group that had separated from Kasakila in the early 1970s, engaged in a sustained, one-sided campaign in which Kasakila males systematically located and overwhelmed every adult male in the Kahama group. It took 4 years. By the end, the Kahama community no longer existed as an independent social unit. The Kasakela group expanded into the vacated territory. The mechanics of how they operated are what separate this from simple aggression. Kasakela males conducted what field researchers described as border patrols, groups of 3 to 8 males moving silently through the periphery of their territory, single file, in a direction away from the core of their range. During these movements, they suppressed the vocalizations they produce constantly in normal circumstances. They checked wind direction. They stopped frequently to listen. The behavior matches the stealth profile used in hunting prey, applied to a social context with a different target category. When they located a Kahama male alone, isolated from any potential support, they engaged immediately and did not disengage until the outcome was complete. Individuals who observed these encounters documented a pattern. Multiple Kasakela males would pin the target, with some holding limbs while others delivered strikes and bites focused on vulnerable areas. The attacks were not brief. They lasted minutes. Research at Ngogo in Uganda, where a larger and longer-running study has been conducted, added a quantitative dimension that the Gombe observations had not fully captured. Researchers at Ngogo documented a chimp community conducting raids over a decade-long period, eliminating adult males from a neighboring group progressively and then occupying the vacated territory. The territorial gain was calculated at approximately 22% of the original range. The researchers noted that the raiding party's decision-making around when to engage appeared to involve numerical assessment. Attacks were initiated consistently when the raiding group outnumbered the target by at least 3 to 1. When the ratio was less favorable, the patrol retreated without engaging. This is not instinctual aggression in the sense of a reflexive response to proximity or threat. It requires tracking the size of neighboring groups over time, assembling a sufficiently large coalition, identifying isolated targets, and suppressing normal behavior, specifically vocalizations, that would compromise the approach. Primatologist John Mitani, who led much of the Ngogo research, described the pattern as the closest documented non-human analog to what, in human contexts, would be called a military campaign. The muscle fiber composition that makes the physical component of this possible has been studied in controlled settings. Chimpanzee skeletal muscle contains a higher proportion of fast-twitch fibers than human muscle, the fiber type associated with explosive, high-force output rather than endurance. Studies comparing the contractile properties of chimpanzee and human muscle tissue have found that chimp muscle generates roughly 1.35 times the force output per unit mass of human muscle, and that the mechanical power output per kilogram is approximately 1.5 times higher. A chimpanzee that weighs 130 pounds generates force at a rate that a 200-pound human cannot match. This difference, applied in a context where multiple individuals are coordinating physical contact with a single target, produces the outcomes that Goodall's team documented. She said the nightmares were about Goliath. Not the mechanics of what happened, but the specific fact that the animals who surrounded him knew exactly who he was and proceeded anyway. She had spent years watching those individuals interact. The relationships she had mapped, the hierarchies she had traced, they did not represent a barrier. They represented prior access. Hippopotamus. Paul Templar was in the water on the Zambezi River in Zimbabwe in 1996 when a hippopotamus took him under. Templar was a safari guide. He was in the middle of leading a canoe tour when one of his clients capsized. He paddled toward the overturned canoe to assist. The hippopotamus that was submerged nearby came up beneath him. In the seconds that followed, Templar was taken into the animal's mouth to approximately the level of his waist, carried below the surface, shaken, released, taken again, and eventually expelled. He survived. He lost an arm. His lungs had been punctured by the animal's lower canines. He described the experience in subsequent interviews and in a memoir with a level of physiological detail that leaves no ambiguity about the force involved. The hippopotamus, Hippopotamus amphibius, is a large, semi-aquatic mammal that spends most of the day in rivers and lakes across sub-Saharan Africa and emerges at night to graze. It is an obligate herbivore. It does not eat fish. It does not eat other mammals. Its digestive system is designed for grasses and aquatic vegetation, and it consumes between 35 and 40 kilograms of plant material per night. During daylight hours, it stays in the water primarily to avoid overheating, since it has no functional sweat glands, and its skin dries out and cracks without regular submersion. None of this produces a calm animal. Hippopotamuses are among the most territorially aggressive large mammals documented by field researchers, and the aggression is not a performance. Adult males patrol and defend stretches of waterway with intensity that does not appear to scale with actual threat level. Animals that enter a male hippopotamus's section of river, other hippos, crocodiles, boats, humans, and canoes, are subject to the same response regardless of context. The hippopotamus does not assess whether the intrusion represents a genuine threat to resources or offspring. It responds to presence. The physical capacity behind that response is considerable. The lower canines of an adult male hippopotamus can exceed 50 centimeters in length, dense, unlapidated ivory that grows continuously throughout the animal's life and is kept sharp by the mechanical abrasion of the upper and lower jaw moving against each other. These teeth have no functional role in feeding. The hippopotamus uses its wide, tough lips to crop grass. The canines are too long and too laterally positioned to be useful for plant material processing. They exist for one application, and that application is other animals. The bite force behind them has been recorded in the range of 1/800 psi, pounds per square inch, in mature adults. For comparison, a large domestic dog produces approximately 300 psi. A lion produces approximately 650 psi. A saltwater crocodile, the animal that shares water with hippopotamuses across much of their range and that researchers generally treat as the dominant predator in African river systems, produces approximately 3,700 psi, but in a narrower mechanical context. The hippopotamus at 1,800 psi generates enough force to compress and fracture the dorsal armor of an adult Nile crocodile in a single closure. Field researchers and rangers working in areas with high hippo populations consistently report one counterintuitive feature of the animal's aggression. It is not consistent with resource logic. Hippos have been documented entering and escalating conflicts in situations where no obvious resource, territory, food, offspring, is at stake. A well-documented incident from Kruger National Park captures this exactly. A group of hippopotamuses converged on a wildebeest that had been brought down by a crocodile at the river's edge. The hippos displaced the crocodile and positioned themselves around the injured wildebeest in a configuration that, observed without context, resembled protective behavior. Tourists watching from a distance reported that it looked like the hippos were guarding the animal. Within a short period, without any change in the wildebeest's behavior or any new external stimulus, the hippos turned and overwhelmed the wildebeest themselves. The crocodile, which had retreated to a safe distance, watched from the water. Researchers reviewing the footage noted that there was no feeding behavior afterward. The hippos moved away. They were back in the river within minutes, returning to the posture of large, slow mammals drifting in the shallows, which is what they look like when nothing has just happened. Templar's account in the interviews that followed his recovery includes a detail that has stayed in the reporting on his case. He described the inside of the hippo's mouth, briefly, before losing consciousness. He came back from that. A significant number of people who have encounters with hippopotamuses in the water do not. Africa-wide incident tracking consistently places the hippopotamus at the top of large mammal attack statistics, substantially ahead of lions, leopards, and buffalo. The animal responsible for the majority of those statistics eats grass in the dark and is sold as a bathtub toy. Pelican, October 2006, St. James's Park, London. A pelican that had been living in the park, along with several others maintained as part of a long-standing tradition dating back to the 17th century, was standing near the lake when a pigeon walked past at the wrong distance. The pelican opened its bill, captured the pigeon, tilted its head back, and began the process of swallowing it. Tourists who were present documented the event on camera. The footage, which circulated widely after being reported by the Daily Telegraph and other outlets, shows the pigeon moving visibly inside the pelican's distended throat pouch for approximately 20 minutes before the movement stopped and the bird continued down. The great white pelican, Pelecanus onocrotalus, and its relatives are specialist fish predators built around a single structural feature that makes them among the most effective aquatic hunters in the avian world, the gular pouch. This is the large elastic bag of skin suspended beneath the lower jaw, familiar from any image of a pelican, which functions as a biological dip net. A pelican hunting fish dives or scoops at the water surface, traps fish and water together in the expanded pouch, then contracts it, forcing the water out through the sides of the partially closed bill while the fish remain inside, before swallowing. The capacity of this pouch in an adult brown pelican has been measured at approximately 11 liters. The stomach of the same individual holds roughly 3 to 4 liters. The catching mechanism holds 3 times what the storage mechanism can accommodate. What fits in the pouch is not limited to what the pelican can ultimately process in one sitting, and the pouch does not appear to communicate that information to the bird in any effective way while the decision to capture is being made. This is why pigeons in St. James's Park occasionally have bad afternoons. The pelican's feeding behavior does not include an assessment of whether the target is a fish. It includes an assessment of whether the target is catchable and fits within the mechanical parameters of the bill and pouch. Pigeons, small ducks, and other birds that share waterfront environments with pelicans fall within those parameters and have been documented as incidental captures at a frequency that is not truly incidental. It is consistent enough across multiple populations that ornithologists treat it as a known behavioral pattern rather than an anomaly. At Dasson Island off the South African coast, and in several documented sites along the Australian coastline, pelican populations have extended this further. In periods when fish availability drops below the threshold needed to sustain the colony, pelicans have been observed organizing movements toward the nesting grounds of neighboring seabird species, cormorants, gannets, and Cape gannets in the South African case, and systematically taking chicks from active nests. The behavior is not solitary. Groups of pelicans move through nesting colonies together, and the taking of chicks is rapid, repeated, and thorough enough that researchers have described it as a significant source of nest failure for the affected colonies during the periods when it occurs. The pelicans involved do not show any behavioral indicators of conflict around this. There is no equivalent of the hesitation or social signaling that researchers have documented in predators that operate in socially complex contexts. The pelican arrives at a nest, the chick is taken, and the pelican moves to the next nest. The structural simplicity of the behavior is, in some ways, its most notable feature. What happened in St. James's Park was documented, reported, discussed in the press, and did not result in any change in the status of the pelicans living there. They remain. Tourists continue to photograph them at close range. The pigeons continue to walk past. Stonefish. A fisherman in Queensland, Australia, stepped off his boat onto what he believed was a rock. The medical report filed at the emergency department that received him describes a patient who arrived in a state of acute distress, escalating rapidly over the 20 minutes between the initial contact and arrival at the facility. The report documents that the patient, at the peak of his pain response, requested amputation of the affected limb. The medical staff did not amputate. They administered antivenom and immersed the foot in water heated to approximately 45 degrees Celsius, the temperature at which the protein-based toxin begins to denature. The patient survived. The nerve damage in the affected tissue was long-term. The reef stonefish, Cynensea verrucosa, is classified by toxicologists as the most venomous fish species currently documented on Earth. This classification is based on the composition and delivery mechanism of its venom, which is not a distinction it earned through any aggressive or predatory behavior toward large animals. The stonefish eats small fish and crustaceans. It ambushes them from the seafloor, using a strike that is among the fastest recorded in any vertebrate. The capture motion completes in approximately 15 milliseconds, too fast for prey animals to initiate an evasion response once the stonefish has begun moving. The venom system plays no role in this hunting process. The 13 dorsal spines, the delivery mechanism, are not used offensively. They are passive. Each spine sits within a sheath of skin, connected at its base to 2 venom glands. When pressure is applied to the spine from above, as occurs when an animal steps on it, the sheath is pushed downward along the spine, compressing the glands and forcing venom up through channels that run along the length of the spine and exit near the tip. The system requires no action by the fish. It requires only contact. The stonefish does not sting. It is stepped on, and the architecture of the spine does the rest. The venom itself, verrucotoxin, along with several associated protein compounds, acts on multiple physiological systems simultaneously. It disrupts cardiac muscle function, produces severe localized tissue breakdown, and activates pain receptors at an intensity that the medical literature describes as among the highest recorded from any biological source. Clinical accounts from treating physicians include patients losing consciousness from the pain response, patients experiencing cardiovascular complications requiring intervention, and patients with tissue damage severe enough that the affected area continues to deteriorate after the initial contact. The breakdown of cellular tissue in the envenomated zone can proceed deep enough to expose underlying structures. The stonefish that produces this is not built to suggest danger. It is the opposite. The camouflage of Sinensia verrucosa is among the most complete passive disguise documented in any vertebrate. The animal's body surface is covered with irregular protrusions, skin flaps, and a texture that replicates the surface of encrusted rock or dead coral in detail sufficient to defeat visual inspection at close range. The coloration varies to match the local substrate. Algae and organic debris accumulate on the skin surface over time, adding a biological layer to the disguise that becomes more convincing the longer the fish remains in position. A stonefish that has been motionless on a reef for several weeks is, to the eye, a piece of reef. In shallow coastal environments, tidal rock pools, sandy reef flats, the kind of areas where recreational swimmers, children, and people in bare feet move routinely, stonefish are present in sufficient numbers across the Indo-Pacific to represent a consistent public health concern in affected countries. Northern Australia, in particular, has established protocols for stonefish encounters that are embedded in coastal emergency medical training. The antivenom for Sinensia venom is the most widely administered fish-related antivenom in Australia. The detail that extends the stonefish's profile beyond simple passive hazard is what happens when the tide goes out and leaves them exposed. Stonefish are not obligate aquatic animals in the way most fish are. Their metabolic rate is low enough, and their respiratory adaptations sufficient, that individuals left out of water by a receding tide can remain viable for up to 24 hours in moist conditions. A stonefish that the morning tide deposited on a rock shelf, or that retreated into a tidal pool that subsequently emptied, is still a functional organism, spines loaded, venom glands intact, sitting in an environment where a person on a morning beach walk might pick it up. The Queensland fisherman who stepped on one was not doing anything unusual. He was stepping off a boat. The stonefish was doing nothing at all. The system that put them both in the same moment was built for something much smaller than a human foot, refined over hundreds of millions of years, and completely indifferent to the difference. Hagfish. July 13, 2017, Highway 101, Oregon. A commercial truck carrying 7,500 pounds of live hagfish was involved in a collision and lost its cargo across the roadway. By the time emergency services arrived, the scene required documentation before it could be explained. A sports car that had been traveling behind the truck was partially submerged in a white, semi-solid material. The road surface was covered. Bystanders described a substance that appeared to have no clear liquid quality. It did not pour, did not drain, and did not respond to the hoses that firefighters initially turned on it. The hoses made it worse. The hagfish, also known as the slime eel, though it is neither a hagfish in the taxonomic sense nor a true eel, had responded to the stress of the impact the only way it knows how. All of them, simultaneously. The Pacific hagfish, Eptatretus stouti, and its relatives, including the Atlantic hagfish, Myxine glutinosa, are among the oldest surviving vertebrate lineages on Earth. Their basic body plan has remained essentially unchanged for approximately 300 million years, which means they were already in their current form before dinosaurs existed, before the continents separated into their current configuration, and before the evolutionary events that produced every other vertebrate group currently alive. In 300 million years, the hagfish has made one significant biological investment: its slime. Along the lateral surface of the hagfish's body, running in 2 rows from head to tail, are approximately 100 glands containing a 2-part secretion. When the glands are compressed, by the grip of a predator's jaws, by the stress of confinement, by any sufficiently intense physical stimulus, they release the contents into the surrounding water. Part 1 is a protein-based mucin, similar in basic composition to mucus. Part 2 is a collection of threads made of an intermediate filament protein, each thread coiled tightly and approximately 15 centimeters long when extended. The 2 components mix in the water column and, within milliseconds, produce a 3-dimensional mesh of protein threads suspended in a hydrated gel. What makes this material remarkable is what it does next. Unlike ordinary mucus, which disperses and dilutes in water over time, the hagfish slime uses the surrounding water as a structural component. It absorbs it. The threads extend, the gel expands, and the volume of the resulting material is approximately 10,000 times greater than the volume of the secreted precursors. A teaspoon of raw secretion produces enough finished slime to fill a 5-gallon bucket. A truck carrying several thousand hagfish, all discharging simultaneously under acute stress, produces a Highway 101 incident. The slime does not behave like a liquid in water. It behaves like a solid. Firefighters using standard pressure hoses found that the water they added expanded the material further rather than washing it away. It required physical removal, scrapers, shovels, applied to a surface that kept reconsolidating as workers moved through it. The sports car was extracted. The road was eventually cleared. The event was reported internationally and generated significant scientific commentary, because the material that caused it is genuinely unusual by any standard applied to biological secretions. The functional purpose of the slime in the hagfish's actual life is defensive. When a shark or large fish attempts to take a hagfish in its mouth, the slime expands inside the predator's oral and gill cavities, physically blocking the respiratory current that passes water over the gills. The predator experiences acute respiratory distress and releases the hagfish, which contracts its body in a knot starting from the tail, working the knot forward along its own body, to strip the slime from itself before swimming away. The hagfish is the only animal known to tie itself in a knot as a functional behavior. It does this routinely. The material properties of hagfish slime attracted the attention of researchers working in defense applications for the United States Navy. The threads that form the structural scaffold of the slime are, weight for weight, stronger than nylon, and comparable to certain high-performance synthetic fibers. More specifically, the capacity of the slime to rapidly and irreversibly clog mechanical systems, pumps, intakes, propellers, was identified as potentially useful in the context of underwater vessel interdiction. Research programs explored the possibility of synthesizing the protein threads artificially as a deployable material. The work continues in various forms. An animal that has no eyes capable of forming images, no true jaw, no scales, no paired fins, and no mineralized skeleton has been producing a material that modern materials science cannot fully replicate for 300 million years, primarily to stop larger animals from eating it. The only reason it is not higher on this list is that it does not go looking for trouble. It just generates it under pressure. Bedbug. The common bedbug, Cymex lectularius, is a small, flat, reddish-brown insect approximately 5 millimeters in length that feeds exclusively on blood. It requires a blood meal to progress through each stage of its development and to reproduce. It feeds only at night, only on sleeping hosts, and it has been living in close association with human beings for so long. Estimates based on fossil and genetic evidence suggest tens of thousands of years, that its behavioral adaptations are calibrated precisely to human sleeping patterns. the timing of its activity, the chemical cues it uses to locate hosts, the suppression of its movement during daylight hours. All of it is structured around a single context: the bed where a person sleeps for 7 or 8 hours in the dark. The feeding apparatus it uses for this is a paired structure called a fascicle, consisting of 2 channels enclosed in a protective sheath. When the bug contacts skin, the sheath folds back and the fascicle extends. One channel delivers saliva, the other extracts blood. The saliva contains a combination of anticoagulants, compounds that prevent the blood from clotting, and anesthetic proteins that suppress pain signaling in the skin tissue around the insertion point. The combined effect is that a bed bug can feed for up to 20 minutes on a sleeping person without producing any sensation that would cause the host to wake. The host provides the blood. The bug provides the pharmaceutical that makes the transaction invisible. After feeding, the bug retreats to a harborage: the seams of a mattress, the gap between a baseboard and the floor, the inside of an electrical outlet plate, any narrow, dark space in close proximity to where the host sleeps. It digests. It waits. It returns on the next feeding cycle, which occurs roughly every 5 to 10 days, depending on temperature and reproductive status. The reproductive biology of bedbugs is where the animal's profile for this list becomes fully established. Male bedbugs do not use the female's reproductive tract for insemination. The female has a functional reproductive system, structurally capable of receiving and processing sperm in the standard way that insect reproduction typically operates. The male does not use it. Instead, the male's reproductive organ terminates in a hardened, asymmetrically curved structure. During mating, the male pierces the female's abdominal wall at a specific point on the right side of the abdomen, injecting sperm directly into her body cavity. The sperm then migrate through the hemolymph, the insect equivalent of blood, to the reproductive organs where fertilization occurs. This process is called traumatic insemination. It is not metaphorical. There is a physical wound produced each time it occurs, and in colony conditions where multiple males are present and reproductive activity is frequent, females accumulate significant tissue damage over their reproductive lives. Immune response to the repeated punctures consumes metabolic resources and reduces female lifespan relative to females maintained without male contact. The female's body has, over evolutionary time, developed a specific anatomical structure in response to this. On the right side of the abdomen, the consistent target site, there is a region called the spermolege, or organ of Berlese. It consists of a depression in the cuticle filled with a high concentration of immune cells and connective tissue. Its only function, as far as researchers can determine, is to receive the male's strikes, limit the spread of damage into surrounding tissue, and mount an immune response to the wound that reduces infection risk. It is an organ that evolved to absorb a repeated injury from the same source. It has no other purpose in the animal's biology. In crowded conditions, which bedbug populations reach rapidly given their reproductive capacity, males also engage with other males, following chemical signals rather than direct visual assessment of potential mates. This has driven additional evolutionary pressure. Males in some populations produce chemical signals that mimic aspects of the female signal profile, which functions as a deterrent to being targeted by other males. The alternative, in the absence of that deterrent, is being pierced. The last figure that situates bedbugs in their proper context. A fed, reproductively active bedbug that subsequently loses access to a host can enter a state of metabolic dormancy and survive without feeding for approximately 400 days. Not weeks, over a year. Sealed in a wall gap behind a baseboard inside the frame of a piece of furniture in a storage unit, present, viable, waiting. The room does not need to be dirty. The building does not need to be neglected. The bug needs a narrow dark space and, eventually, a person. The person does not need to do anything to become involved. They just need to go to sleep. Cat. In 1894, a lighthouse keeper named David Lyle was posted to Stevens Island, a small, steep, isolated island off the northern coast of New Zealand's South Island. He brought with him a domestic cat named Tibbles. The island was remote enough that it had no permanent human population and had never been settled. Its native fauna had evolved in the absence of mammalian predators. One of the species living on Stevens Island was a small, flightless wren, the Lyle's wren, later formally designated Traversia Lyalli, that had existed on the island in complete isolation. It could not fly. It had no behavioral response to mammalian predators because it had never encountered one. Tibbles found them at night when they were active, moving through the low vegetation on the ground. Over the following months, Tibbles brought specimens to Lyell, who recognized they were scientifically significant and sent several to the ornithologist Walter Buller and to the Natural History Museum. The scientific community became aware of the species and its existence on Stevens Island at approximately the same moment the species ceased to exist on Stevens Island. The last confirmed specimen was collected in 1895. The Lyell's wren was discovered and made extinct within the span of a single cat's active period on a single island. The historical record is somewhat more complicated. There may have been feral cats on the island in addition to Tibbles, and lighthouse construction itself disrupted the habitat. But the core fact is not disputed. One domestic cat, operating freely on an island with no prior mammalian predator exposure, was the primary driver of the elimination of an entire vertebrate species from the planet in less than a year. This is the species that researchers at the University of Georgia decided to study more systematically in 2011, using miniature video cameras attached to the collars of free-roaming owned cats in suburban environments. The KittyCams project produced footage of 55 cats over extended periods, capturing their behavior during the hours they spent outside without their owner's knowledge. The footage confirmed several things that researchers had inferred from prior studies, but had not directly documented at this scale. The cats in the study hunted consistently. The average cat in the sample made multiple capture attempts per week during outdoor time. But the detail that stood out in the analysis was not the frequency of captures. It was what happened after them. In 72% of all successful captures documented in the KittyCams footage, the cat did not feed on what it had caught. The animal was brought down, and then it was abandoned, left where it fell, or carried back to the residence and deposited at the door. In the remaining 28% of cases, the capture resulted in partial consumption. Full consumption was the least common outcome of a successful hunt. The behavior that produces this, capturing and releasing, or capturing and abandoning without feeding, has been studied in the context of feline predatory neurology. The drive to hunt in domestic cats is not tightly coupled to hunger in the way it is in obligate wild predators, where the energy cost of hunting must be offset by the caloric gain of eating. In domestic cats, which receive food independently of hunting success, the predatory sequence, stalk, pounce, capture, produces neurological reward through the behavior itself. The completion of the sequence is the point. What happens to the prey afterward is a separate question, and for a well-fed cat, often an unresolved one. The tongue that the cat uses to process a capture it does eat is worth describing on its own terms. The surface of a cat's tongue is covered with small, backward-curved projections called filiform papillae, made of keratin, the same protein that forms fingernails and horn. Under magnification, these papillae are hollow, drawing saliva into their structure and releasing it during grooming. They function in terms of texture as a biological rasp, a surface that strips material from whatever it contacts rather than simply moving across it. A cat grooming its own coat uses these structures to detangle and clean fur. A cat processing prey uses the same surface to strip tissue from bone with a mechanical efficiency that requires no additional tools. The aggregate impact of this specific animal, the domestic cat kept as a companion in homes globally, has been calculated in population-level studies with figures that require a moment to absorb. In the United States alone, free-roaming domestic cats are estimated to be responsible for the removal of between 1.3 and 4 billion birds annually, and between 6.3 and 22 billion small mammals. The range is wide because precise measurement is methodologically difficult, but the lower bounds of both estimates are already larger numbers than any other human-associated source of direct wildlife decline. The International Union for Conservation of Nature lists the domestic cat among the 100 most damaging invasive species on Earth, a list that otherwise consists primarily of agricultural pests, disease vectors, and organisms introduced deliberately for ecological management purposes that subsequently exceeded their intended scope. The cat responsible for the Stevens Island extinction was living in a lighthouse keeper's home and being fed regularly. The cats documented in the KittyCam study were returning from their outdoor time to bowls of commercial food in climate-controlled houses. The hunting is not contingent on need. It is simply what the animal does when the opportunity presents itself, because the opportunity and the neurological sequence it triggers are enough. Prairie dog. The black-tailed prairie dog, Sinomys ludovicianus, is a rodent. It weighs between 0.7 and 1.4 kilograms. It lives in burrow systems called towns on the short grass prairies of North America, eats grasses and forbs, and communicates through a system of vocalizations that researchers have studied extensively. Prairie dogs produce calls that appear to encode specific information about approaching predators, including descriptors of size, shape, color, and speed of movement. Their alarm call system is among the most informationally dense animal communication systems documented. They are strictly herbivorous. This is established in every introductory zoology text that covers North American mammals. They eat plants. They have the digestive system of an herbivore. Their teeth, large, chisel-shaped incisors and flat molars, are designed for cutting and grinding plant material. The prairie dog is not a predator. In 2016, ecologist John Hoagland published a study in the Proceedings of the Royal Society that required a significant revision of that picture. Hoagland had been studying black-tailed prairie dog colonies for decades at field sites, including Wind Cave National Park in South Dakota. Over the course of his research, he and his colleagues documented something that had not been recorded in prior studies of the species: prairie dogs pursuing, catching, and ending the lives of Wyoming ground squirrels, a smaller rodent species that shares the same prairie habitat. The prairie dogs were not eating what they caught. The ground squirrels were pursued, brought down with bites directed at the skull and cervical spine, the incisors that normally process grass roots applied to the vertebrae of another animal, shaken and left in the grass. The bodies were not consumed. They were not cached. They were abandoned where they fell. Over 5 years of systematic observation, Hoagland's team documented 163 individual acts of this kind, carried out by prairie dogs across multiple colonies. They identified the individuals responsible, tracked them across seasons, and found that the behavior was not uniformly distributed through the population. Specific females were responsible for the large majority of recorded incidents. These individuals were not behaviorally unusual in other respects. They foraged normally, maintained burrow systems normally, interacted socially with other prairie dogs normally. They also, at intervals, located ground squirrel pups near their burrows and eliminated them. The functional logic that Hoagland proposed was resource-based. Ground squirrels eat the same grasses that prairie dogs require, and removing them from adjacent territory increases the availability of food plants for the prairie dog's own offspring. The herbivore was not hunting for protein. It was clearing competition for its primary food source using the only tools its body has. The same incisors designed for roots and stems, redirected at a different target. The reproductive data that Hoagland collected made the pattern harder to dismiss as incidental. Females that had documented records of ground squirrel elimination produced, on average, litters approximately 1.5 to 3 times larger than females with no such records, when controlling for other variables. They also showed higher rates of offspring survival through the first year. The behavior that is outside every prior description of the species correlated consistently with improved reproductive outcomes for the individuals who performed it. Hoagland documented one female over a single morning that located and eliminated 7 consecutive ground squirrel pups from burrows adjacent to her own. After each one, she returned to her own burrow entrance, oriented for several seconds, and then moved to the next. No alarm was raised. No other prairie dog in the colony reacted to what she was doing. She had been, by Hoagland's data, one of the most reproductively successful individuals in the colony for multiple consecutive seasons. She was eating grass when his team arrived that afternoon. The textbooks that described prairie dogs as strictly herbivorous were not wrong about what they had observed. They were incomplete about the full range of what the animal does when the conditions are right and the cost is low and the grass belongs to whatever happens to be living next door. Hoagland published the data. The species remains classified as herbivorous in most general references. The ground squirrel population near the study colonies remains lower than baseline models predict it should be, in a way that took 5 years of camera traps and direct observation to explain. 163 documented cases, 1 study, 1 ecologist. The prairie dog didn't change, the record of it did.